left off talking about the flagella and the cilia, right? That last little bit here. I know we kind of covered this already, but I just wanted to review it. So flagella, they're normally longer, they're singular or in pairs typically, and they have this 9+2 microtubule arrangement, which I will show you in just a couple slides. Cilia tend to be shorter and tend to cover the whole surface of the cell. All right, so this movement is propagating of a bend. All right, so we talked about the motor protein, where the protein was stationary and the filament moved from underneath of it. And we talked about when the filament was stationary and the motor protein moved. All right, think about the life of John, that motor protein that we saw. This is where we have the propagation of a bend. And then I think this video, this is just an image showing you flagella, longer, singularly, cilia here. But I have a video that I think is going to help you visualize it a little bit better. I still haven't figured out why the audio is not working. So we're going to give this a shot.
Motile cilia and flagella are hair-like appendages of cells that generate fluid flow along tissues or allow cells to move through their environment. To better understand how these appendages generate motility, we flash froze sea urchin sperm cells while their flagella were actively beating, and then directly visualized the molecular machines inside the flagella through cryo-electron tomography. Thousands of motor proteins, called dyneins, are arranged regularly along the microtubule scaffold. Through classification, we identified several different functional states of the dyneins as they step along the microtubules. We demonstrate a detailed stepping mechanism. From the relaxed post-power stroke state, the dyneins release from the microtubule, swing forward, and rebind to the microtubule. During the power stroke, the cargo is pulled forward by the action of dynein. As long as ATP is present, dyneins continue to walk along the microtubule. However, if dyneins receive inhibitory signals, they can be kept in intermediate states that resemble the post-power stroke state, leaving them unable to generate motion. Our direct visualization of specific dynein conformations on either side of the flagella suggests a switch inhibition mechanism generates the flagellar beat. As one side is inhibited, the other side is active. In our model, regulatory signals result in inhibited dyneins on one side. This allows dyneins on the opposite side of the flagellum to walk along the microtubules. Molecular linkers resist this sliding, which causes the flagellum to bend in one direction. By periodically switching the side of dynein inhibition, the flagellum alternates the direction of bending, resulting in the typical whip-like motion. The propagation of dynein inhibition in different regions of the flagellar wave along the length of the flagellum leads to the characteristic beating motion. The dynein inhibition may occur in an all-or-none fashion, similar to an action potential, which could explain how cilia and flagella can switch their bending direction back and forth in less than 1/100th of a second and continue beating for days.
All right, so I thought that video demonstrated this bending movement and how that can this bend and how that can result in movement a little bit better than I could just by showing you images. Let's see here. But there is a little bit of terminology that they used in there that I also want you to be aware of. So we're talking about a dynein. That's that specific motor protein that's being used. That's the most typical in these flagella and these cilia. We have the dynein here in our image, and in our little cartoon, it's these yellow squiggly lines here. Notice they're only attached to one side of these green microtubules. All right, it's attached to one side. The other side does not have dynein attached. All right, that is so it can go and walk along that unattached side, causing that bend. As a whole, all right, this cross-section would be an axoneme. All right, so that's the technical terminology for this cross-section is the axoneme. That's all the internal structures of the microtubule. It includes the dynein. It includes these linking proteins, which kind of links everything together. In this array, like I said, they typically have a 9+2 array. So this is the explanation of that here. So we have the 2 in the middle, and then we have 9 sets of a doublet, or 9 sets of 2 around the outside. So that's what that 9+2 array refers to. You can have different arrays, all right, but they're just less common. And finally, we have a basal body. All right, the basal body is simply where the microtubules are going to attach to the main part of the cell. All right, any questions about flagella, cilia, anything in that realm? All right, just as a review, I put this picture up here to show you, we have multiple parts of the cytoskeleton. They all kind of interact together, and they all influence different parts of what's going on in the cell. What might be cell shape and motility, might be anchoring organelles, or it might be providing a track for movement. All right, additional resource. This is a little bit more in-depth in microscopy. Thought it was kind of cool, but it is a little bland, so I don't blame you if you don't want to watch it, but if you're interested, that's a great video. Let's move on to our other lecture here. All right, so now we're going to dive a little bit more into the organelles. So we talked about the cytoskeleton a little bit here. Let's talk about cell structure and organization. All right, these are the key concepts we'll be going over today. But let's start with the endomembrane system. All right, so endo meaning inside. All right, so we're talking about all the membranes that are inside of the plasma membrane. All right, so all these organelles inside of our plasma membrane are connected through this endomembrane system, whether the membranes of the organelles are connected directly or indirectly. All right, so sometimes they're physically connected. Sometimes they might be connected through vesicles, all right, which are basically these spheres that are going to move between the different organelles. Now, many of these organelles are going to work together, all right, as we'll see in a bit here. They all kind of work with each other. Some of them might synthesize proteins, some of them might store proteins, some of them might ship those proteins off to different areas. So they all work together in conjunction here. All right, so what is included in our endomembrane system? Well, we have a lot of things here. All right, we have the nuclear envelope, we have the endoplasmic reticulum, the Golgi apparatus, lysosomes, vacuoles, and also the plasma membrane. All right, so we're going to go over each of these individually. The plasma membrane we're not going to get to until Wednesday. And kind of as a little quick note here, all right, so it was thought that endomembrane, meaning inside of the plasma membrane, that this membrane system was excluded from the plasma membrane, all right, that it was just all the membranes of the organelles, and then you had the plasma membrane. But it turns out the plasma membrane is pretty integral into this endomembrane system as well. So now it's included as part of this endomembrane system. All right, the organelles are working together, and they're also communicating with the plasma membrane. All right, let's start by talking about the nucleus. All right, I think this is one of the most prominent features of the cell here. So the nucleus contains that DNA and directs protein synthesis by making messenger RNA. We're not going to go in depth on that process today. That'll be a couple weeks down the road here. What I want you to get from this is that the nucleus contains DNA, directs protein synthesis. All right, pretty crucial in that aspect. Kind of the structures I want you to be aware about in the nucleus is that it has this double membrane system. All right, so it has two membranes surrounding it. All right, the outer membrane is actually continuous with the endoplasmic reticulum. All right, another organelle we'll talk about in just a minute here. All right, and then we have these pores. All right, so these nuclear pores are going to provide a passage between the inside of the nucleus and the outside of the nucleus. All right, so the nucleus isn't just all self-contained. We have things moving in and out of the nucleus, whether it might be proteins, it might be RNA moving back and forth, it might be ribosomes. All right, everything inside of their double membrane system, not part of the endomembrane system. All right, so we're only talking about kind of the membranes here. All right, so in the nucleus, we have this region called the nucleolus. All right, now the nucleolus is where we have ribosomal subunits that are going to be assembled together. All right, so talking about ribosomes, let's move on to them here. So ribosomes, they're important for protein synthesis. All right, and they consist of these two subunits. And we can see these subunits in this cartoon drawing below here. It's typically a smaller subunit and a larger subunit. All right, one big, one small, and they're going to contain one or more RNA molecules as well as several different types of proteins. All right, now these subunits, they're assembled in that nucleolus there, and then they're moved outside of the nucleus via those nuclear pores. All right, let's see here. Oh, I have another side of ribosomes. All right, so they're synthesized from ribosomal RNA. All right, that's produced in the nucleolus. All right, again, we're not going to go too much in depth into RNA right now. We might have cells that have to make a lot of protein. They might have a lot of ribosomes. All right, ribosomes, site of protein synthesis. Makes sense. If you need more proteins, you need more ribosomes. Now, these ribosomes can be kind of suspended in the cytoplasm, or they can be attached directly to the endoplasmic reticulum. I kind of show an image here. These are just kind of suspended in the cytoplasm. And if we look at our endoplasmic reticulum, all these kind of bumpy shapes here, those would be the ribosomes attached. So, endoplasmic reticulum, all right, it's kind of this network of membranes. And they form these flattened, fluid-filled tubules, all right, that we refer to as cisternae. All right, let's see, is it written on the slide? Yes, it's right down here. So that's a cisternae. I'm not sure if I'm pronouncing that totally correct there. But basically, this is referring to those flattened tubules. All right, and we're not only going to see that in the endoplasmic reticulum, we're going to see them in other organelles as well. So it's not specific to the ERs, all right, but it's describing what the ERs look like, or their structure. All right, so endoplasmic reticulum, we have two basic types, all right, the rough ER and the smooth ER. All right, the rough ER is characterized by having ribosomes, all right? They have ribosomes in and embedded within their membranes. We can see that here. All these little ribosomes are attached, all right? Let's see, they are important in protein synthesis because they have those ribosomes attached. They are also important in protein sorting, all right? So directing where proteins might go after they're synthesized. They're also important in inserting proteins into membranes, all right? So if we have our plasma membrane, we might have a protein channel that allows certain molecules in or out, all right? The rough ER is where those proteins will get inserted into the membrane and then shipped off to the plasma membrane. All right, we also have attachment of carbohydrates to different proteins. All right, this is called glycosylation. All right, this can also occur in the rough ER. All right, it can also make additional membranes for itself, and essentially, it grows. All right, now contrasted to that smooth ER, all right, we're lacking ribosomes at this point. All right, so not very important in protein synthesis, but they are important in the synthesis of other macromolecules. Lipids, oils, phospholipids, steroids, all right? They also play a major component in helping to store calcium ions. All right, so, don't know why that popped up. All right, just as kind of like to drive home that process that we can synthesize proteins and then ship them off to other areas. All right, we have our ribosome here. We know it's the rough endoplasmic reticulum because it has this ribosome attached to it. It's synthesizing a polypeptide. All right, we're adding on a sugar chain or a carbohydrate. All right, so it's now become a glycoprotein through the process of glycosylation. And then essentially this vesicle is going to bud off of the endoplasmic reticulum and be shipped to another part of the cell. Whether it's another organelle, whether it's the plasma membrane, totally depends. All right, but this is kind of the basics of how that's going to happen. All right, so the next organelle, kind of going out from the nucleus here, is the Golgi apparatus. All right, so the Golgi apparatus can be known by many different names here, but they're stacks of flattened membrane-bound compartments. All right, again, this is where we get that term cisternae. All right, it's flattened membrane-bound components. Now, this Golgi apparatus is in blue here. All right, has a lot of important functions, some of which is transporting different molecules. They can transport it between their individual stacks of these flattened compartments here. They can transport it to the plasma membrane or a different organelle. All right, so they have these three kind of overlapping functions. The first is secretion that we'll talk about. And this is where we're going to package different materials into these secretory vesicles that are going to fuse with the plasma membrane and release it to the outside of the cell. All right, so we can kind of see that here. We're budding off this vesicle. All right, it's now a free vesicle here, moving towards the plasma membrane, fuses with the plasma membrane, and empties out its contents. All right, so we can secrete things outside of the cell. We also have processing that can occur. All right, so that's where we might modify proteins. All right, we might be adding in carbohydrates, we might be adding on lipids to proteins, but we're processing them. In this processing stage, we can also get something called proteolysis. All right, kind of a mouthful there. It's basically where we have these enzymes that are going to cut proteins that are larger into smaller enzymes. All right, and this is important with some molecules such as insulin. All right, when insulin is first produced, it's a larger protein, and we need to cut it down into smaller subunits to get insulin. All right, so this is important in the processing step. And then we also have protein sorting. So we're sorting out different proteins from each other, depending on what they are or where they need to go. All right, so I kind of think of this as like the postal service system of the cell. All right, we also have these lysosomes. All right, so lysosomes are very small organelles, and they break down macromolecules. All right, so they contain an acid hydrolase, which is an enzyme that's going to perform hydrolysis. All right, so if we remember what hydrolysis is, that's basically we're throwing a water atom or water molecule at a larger macromolecule and breaking the bonds. All right, we're using water to break a bond. Now this works best at a very low pH, all right, and that's why we have these acid hydrolases, acid enzymes. Now we can have many different types of these enzymes to break down different macromolecules. All right, so it's not just one type of enzyme they carry, they can have multiple enzymes. And we can digest molecules that have been taken into the cell by endocytosis. All right, so endocytosis. This is where we're basically taking in some sort of food molecule. All right, it's been preliminarily broken down into these smaller components, goes into this food vacuole. This lysosome will come and attach to the food vacuole, release the enzymes, and digest these molecules. All right, so they play an important role in digestion. All right, so speaking of vacuoles, let's move on to our next organelle that we're going to talk about, which are vacuoles. All right, so vacuoles are these larger, well, they can be these larger membrane-bound structures. They were once thought of as being empty. I mean, if we look at them here, that looks pretty empty, right? But they actually contain a fluid and sometimes solids. Like we just saw, it might be a food vacuole, might contain some solids that are a type of food. All right, and we have different types of vacuoles that can be found in different types of cells. So in plants, for example, we have a central vacuole. All right, now plants use this central vacuole for storage and support. All right, it's typically very, very large. I think the statistic is it takes up like 80% of the entire cell. All right, it's very important in storing excess sugars or excess water, especially if you get like a succulent plant. They're going to have very large vacuoles with a lot of extra water in them. They're also important for support. All right, so because they are able to take up all this extra water, they apply pressure on the outside plasma membrane and help provide rigidity in these cells. All right, this is what we call turgor pressure. Don't think we're going to get into it in this lab or this lecture here, which is kind of disappointing because I love talking about turgor pressure. All right, but it's providing pressure that gives the cells rigidity. All right, now these central vacuoles are only going to be really found in plant cells. All right, this is different from these contractile vesicles. All right, so these contractile vesicles are going to be used to expel excess water. All right, so different cells might want to maintain their shape, all right, and maintain their shape and their size. And water is always going to be entering in through this diffusion. All right, so basically these contractile vesicles are storing that water and expelling it back outside of the cell so that cell can maintain its shape and size. All right, the last one we'll talk about is the food vesicle. All right, this is where food is taken into the cell, stored in these vesicles for other organelles or other molecules can come in and start to break down those food. All right, so think about that forming with lysosome here, like we talked about on that last slide. All right, I believe this is the last one we're going to talk about in terms of organelles, the peroxisomes. All right, so the peroxisomes, they're catalyzing detoxifying reactions. All right, and when I say catalyze, they're essentially speeding up these reactions. All right, so we have toxins that can enter into the cells, and these peroxisomes are responsible for breaking down those molecules, typically by either removing hydrogen or adding oxygen. All right, now this is going to create a byproduct of hydrogen peroxide. Now, hydrogen peroxide can be toxic in the presence of certain atoms. All right, so we also have to break down this hydrogen peroxide, and that's where we have these enzymes that will break down the hydrogen peroxide further into oxygen and water. All right, so think about liver cells. They're important in taking care of toxins. They're going to have a lot of peroxisomes. All right. I was going to include a group question here, but I decided not to because I have a worksheet for you guys instead. Is there any questions so far, though? I know it's a lot of information being thrown at you. All right, then I'm going to keep lecturing. All right, I don't have too much more for lecture, and then we'll get into a group exercise here. So we talked about the endomembrane system, right? But we also have these semi-autonomous organelles. All right, so these are organelles that can grow and divide almost independently to reproduce themselves. All right, so in cells, this would be mitochondria and chloroplasts. All right, now they're not completely autonomous. All right, they still depend on the cell for synthesis of their internal components. All right, so they still need building blocks from the cell in order to grow and divide. All right, now notice we have. Oh, I thought I had this image on the next slide as well. But we have mitochondria in animal cells. Plant cells can have mitochondria and chloroplasts. Animal cells are not going to have chloroplasts. All right, so these two different types of organelles are going to carry out reactions that'll help with energy conversion. All right, so mitochondria, they're going to carry out cellular respiration. We're turning that food into ATP. All right, so we're not creating energy per se, all right, but we're creating the molecule that can be used for energy. Now, chloroplasts are basically doing the same thing, but for plants. All right, we're taking in light energy, and we're converting it to sugars. All right, we're creating these molecules that can be used as energy in other reactions down the road. So mitochondria, all right, they have these specific structures to them here. They have two membranes, an outer membrane and an inner membrane, and then they have this intermembranous system or space in between. All right. Now notice that this inner membrane is highly folded. All right. This gives it a lot of surface area. And this is important because in this membrane system is where ATP is created. So if we have more surface area, we can create more ATP. All right, so highly advantageous. All right, we also have its own DNA, these mitochondrial DNA. It has its own ribosomes as well as enzymes that it can use for respiration. All right, so it's essentially a self-contained system that can almost exist independently on its own. All right, because it has its own DNA, all right, it can divide on its own. So we call this binary fission. All right, both chloroplasts and mitochondria will do this. Essentially, the mitochondrial chromosome here, it only has one chromosome, is going to divide. All right, and once that divides, we can divide the cell into two. Again, we're not going to get into the details of this here. And then we have two mitochondria. All right, now this is important when we think of cell division. We want to make sure when a cell divides, each part of that cell is going to be able to have mitochondria. But it's also important if we say we start using more energy. All right, maybe you just got into running and your muscles are using more energy. these mitochondria are going to divide and replicate in order to provide more ATP to supply those cells with energy. All right, so it could be we need to make more of these for cell division or as needed. Chloroplasts, on the other hand, they're capturing light energy. All right, and we're using that light energy to synthesize molecules such as glucose. All right, and these glucose molecules can be the building blocks for other sugars down the road here that can be used as energy inside of the plant. All right, so they're found in plants, they're found in algae. Anything that can photosynthesize is going to have chloroplasts. All right, they also have an inner and an outer membrane. They also have a third membrane called the thylakoid membrane. And I'll show you a picture of this in just a second here. But this membrane plays an important role in photosynthesis. They also contain their own DNA. They also divide via binary fission here. All right, so again, we have an outer membrane, an inner membrane, and we have this third membrane system called the thylakoid. All right, they can be stacked up here, and this is important for photosynthesis, all right, which we will talk about in a few weeks, so I'm not even going to try to go into those details here because it'll just be overwhelming. All right, so I kind of had you guys watch that little video that kind of got into this idea of the endosymbiosis theory. All right, so why do these organelles have their own DNA? All right, how did this happen? So in order to understand this, we have to understand endosymbiosis. All right, so it's a symbiotic relationship. Basically, two organs or organisms are living together in harmony. Smaller species can live inside of the larger species here. Think about the bacteria that live inside of our gut. All right, they're living inside of us. And the endosymbiosis theory basically says that mitochondria and chloroplasts originated from bacteria that started outside of a cell and then started living inside of the eukaryotic cell and found it to be beneficial for both of them. All right, so just take a quick look here. All right, we have this Propylbacterium or the Cyanobacterium, which is thought to be the evolutionary predecessor of the chloroplast. All right, it made its way inside of the cell. Once it got inside of the cell, it found that it was very beneficial. It was receiving nutrients and building blocks from the cell that it didn't have to synthesize on its own. All right, in return, the cell enjoyed having it because it provided a lot of energy for other processes inside the cell. So over time, through evolution, they evolved to coexist together. All right, so basically we have these four interacting parts of this eukaryotic cell that we have gone through so far. We're going to elaborate on this a little bit more on Wednesday. I did want to throw in just a little extra that I know wasn't required in the reading here, but I think it's going to help you guys understand a little bit more what's going on here. I'm just going to quickly go through this. All right, so outside of the plasma membrane, the cell does not just stop. Right? There's more to our cell. All right, it's actually kind of like this meshwork of macromolecules outside of the cell. It's the extracellular matrix in animals and cell walls in plants. All right, so the extracellular matrix in animals, they can play some really main roles, all right, such as providing strength or structural support. Maybe they're helping organize cells next to each other or helping a cell signal to one another. All right, so they play extremely important roles. They should not be forgotten about when we talk about the cell. And again, it's kind of like this meshwork here. All right, we don't want it to be totally solid because we want water and other molecules to get through to that plasma membrane. Same goes for the plant cell walls. All right, they provide strength and they provide rigidity. Plant cell walls actually have two cell walls. The primary cell wall, which is more flexible to help it grow, and a secondary cell wall, which is super rigid and what is found in wood. All right, here we have our plasma membrane. We have our primary cell wall, and then in between we have our secondary cell wall. All right, and again, it's not just solid like this image might depict here. It's this network of different macromolecules intertwined together to allow substances to move towards that plasma membrane. All right, so I'm going to stop lecturing there, and the rest of the time we're going to work on this activity as a group. And it's just asking you questions about what we've learned about so far and kind of making you think a little deeper. Apply these concepts, not just write down definitions. Now, when I printed this out, I thought I was going to print double-sided. It did not. So there are 2 sheets that you'll need to pick up. So make sure you get both of them. I'm just going to split this in half, and we will start them on opposite ends of the room here. Get the sheet, work together as a group, ask me questions as you go along, make sure you're understanding this. When you are done, you can leave. Prep for Wednesday, I didn't put in a prep for Wednesday. All right, I think we're going to have a lot of stuff to do on Wednesday as is, but know that there is a quiz 2 on Friday. All right, you do have that assignment 1 that is due on the 27th, and the exam is coming up really soon. All right, so there might be no prep, but you still should be working on these things.